Hi, my name is Justin, and I'm an undergrad student at the Ciernia Lab. Our lab is particularly interested in how early life changes in the brain, especially genetic changes, can alter behavior later on in life. And personally, I'm more interested in the bioinformatics side of things, so a lot of what I do tends to be more on the computational side. Uh, a lot of my previous work has involved interrogating changes in gene expression after our experiments. And so, an example of what I might do would be taking an RNA sequencing dataset and utilizing a variety of tools, primarily R, but sometimes also the command line, and using a lot of coding, a lot of data crunching, sometimes on my laptop or on a web server. I would then try and turn it into something meaningful. So, for example, I might cluster the samples to try and identify similarities between them, or try and identify patterns in gene expression, or just identify genes of interest, and other fun things we can do with visualization. And so, through studying these changes in the patterns of gene expression within the brain, we can potentially identify what's driving these alterations in behavior, or potentially learn more about what's underlying more complex neurodevelopmental disorders and diseases.
Hi, I'm Megan, and I'm a postdoc in the Ciernia Lab. I study BAF53B, which is part of the switch-SNF chromatin remodeling complex. This complex is important for altering DNA accessibility and, in turn, gene expression. What's unique about BAF53B is that it's expressed exclusively in neurons, and humans with mutations exhibit autism spectrum disorder-like symptoms, and in mice, it's really important for neuronal branching as well as synaptic plasticity and memory formation. Even though BAF53B seems really important in neurons, its function in the individual neuronal subtypes has never been investigated. So for my project, I'll be deleting it in either excitatory or inhibitory neurons, and then assessing mice for autism spectrum disorder relevant behaviors. Both male and female juvenile mice will be assessed for ultrasonic vocalizations, juvenile play, and developmental milestones. And then these same mice as adults will be assessed for anxiety, repetitive behaviors which can be indicative of autism spectrum disorder, social behavior, as well as memory formation.
Hi, I'm Olivia. I am a fourth year behavioral neuroscience student and a directed studies student in the Ciernia Lab. And so far, my work with the lab has really involved piloting out all of the behavioral tests that we're now able to run in the lab. And so some of the behaviors that we were looking into tests for include anxiety, repetitive behaviors, social behaviors, learning and memory, and depressive-like behaviors. So as an example, one of the tests that we piloted out this summer was called the 3-chamber test. So this is a test of social behavior in mice, and essentially what it involves is that you have a box that is divided into 3 different sections, and you have one of the sections that just has an object, and the other section has a mouse. And you place the test mouse in the center, and you allow it to go wherever it wants. And the expected behavior, or the normal mouse behavior, would be to spend majority of its time in the chamber with the new mouse, because mice are social creatures and they tend to exhibit high levels of social behaviors. And so by the end of the pilot experiment, which was really just working out the logistics of each of these tests, sort of like what equipment we needed and exactly how to run these tests. So by the end of the pilot, we had this huge behavioral assay of all these different tests that we're now able to run efficiently in the lab and use throughout various experiments within the lab.
Hello everyone, my name is John and I'm a co-op student from the Ciernia Lab. My work involves designing automated data pipeline for mouse behavior analysis. As you can see on the top left corner, I am using a software called Deep Lab Cut, which allows you to track different body parts of an animal. And with the output, I can do further data analysis, such as looking at change in distance traveled over a period of time. Going forward, I'm excited to use machine learning to classify different mouse behaviors. And if you have any questions or want to check out the source code, you can visit the Ciernia Lab GitHub page. Thank you.
Hey everyone, I'm Jen and I'm a PhD student in the Ciernia Lab. My work focuses on understanding how the immune and nervous systems interact to impact brain function and behavior. I look specifically at the cell type microglia, which are traditionally known as the brain's resident immune cell. Immune cells can adapt to a changing environment by enhancing or repressing their responses to future immune challenges, a process known as innate immune memory. Prior studies point to epigenetic modifications as key regulators of innate immune memory in peripheral immune cells, but these mechanisms aren't nearly as well understood in microglia nor in the context of the brain environment. In my research, I use a variety of approaches, including microglia morphology analysis, epigenetic sequencing, and behavioral testing of genetically manipulated mice to characterize how epigenetic mechanisms previously identified in peripheral immune cells might regulate microglial responses to shape immune memory in the brain.